Now we are starting here. Classification of living beings. Here I want to introduce some of the concept or basis of classification. First, we have to discuss about cell type. Now we are aware that every living being is made up of cell. Maybe possible some living beings are unicellular in nature. Uni means one. That means they are having only one cell, like you are aware of amoeba. Then there are multicellular animals also. But certain living beings, say for example, if I want to talk of viruses, then we are considering viruses on border of this system, because we can't consider them as living as well as non-living. Reasons we'll discuss later. So here it's very clear for you that we are talking about life system, and in that we are talking about first cellular structure. Basically, two types of cells are observed. They are called as prokaryotic cell and eukaryotic cell. So first I should give here a clear-cut idea that cells we are dividing into two parts. One that is called as pro, and second that is called as eukaryotic cell. Why this type of classification is required? Let us check out. Prokaryotic cell, as name indicates, it is pro. That means it is prior or earlier form. They are not having departmentalization. Suppose you are visiting any institute. If it is small institute, there is no departmentalization, no cabins are there. All things are happened in only one single office. But if you are visiting out huge institutions, then there is departmentalization. We are having office area separate. Suppose that is college. Then laboratories are separate, lecture rooms are separate. All these governing bodies like principal, they are having separate office. Supervisors, they are having separate cabins, and like that. So here I should consider this is pro as primitive one, but really speaking, I don't feel there is any primitiveness in this part. Anyway, we are talking of now prokaryotic cell. There is a capsule. Inside this capsule, there is a material we are calling that as cytoplasm. So that cytoplasm is present. You are aware that whatever inside this capsule, we are calling that as cytoplasm, or many times we refer as protoplasm also. Protoplasm means whatever there inside the cell, that is called as protoplasm. Whereas cytoplasm, that means a jelly-like liquid that is present inside this tube or a capsule, that is called as cytoplasm. And organelles, instead of organelles, certain bodies, they don't have any membrane. They are floating anywhere in the cell. So this type of cells, we are calling them as prokaryotic cell. So you will not get any definite organelles. That is the characteristic of prokaryotic cell. So bacteria, best example, that is bacteria are belonging to this type of cell, that is called as prokaryotic cells. Bacteria, maybe they are autotrophic in nature, maybe they are heterotrophic in nature. You are aware of the word, but otherwise I should tell you, autotrophic means they prepare their own food with help of photosynthesis. Many times bacteria are carrying out another type of synthesis, that is called as chemosynthesis. So bacteria, they are either photosynthetic or chemosynthetic. Then heterotrophic. Heterotrophic means they are not preparing their own food. They depend on others for getting their food. So they are called as heterotrophic bacteria. So here we are saying this is as prokaryotic cell. What is the predominant characters of this prokaryotic cell? That is, these cells are not having membrane bound organelles inside the cell. Only capsule is there. Cell size is relatively smaller. They are either solitary or colonial in nature. solitary, that means living their own, single. Whereas colonial, they are having groups and living in the form of groups. So this is about prokaryotic cell. Now eukaryotic cell, I should divide into various categories, but here I am dividing only in two categories first. They are called as plant cell and animal cell. Again, in every category we have somatic cells and gamete cells. They are also separate, but right now we are focusing primitive type of classification. So here we are talking of plant cell and animal cell. So this is prokaryotic cell. Now we are focusing on eukaryotic cell. In case of eukaryotic cell, we are going first to explain animal cell. In this animal cell, there is again some here we are having capsule. So similar, here we are having a capsule, but it is not called as capsule, it is called as plasma membrane. Let me clarify more here. You can compare egg. Egg is also cell, but it is special form of cell. We are calling that as gamete cell. Here we can consider egg as cell because that cell we can observe with our eye. So it is not necessary that we have to observe through microscope. So these cells are observed only under microscope. But egg cells, being special cells, they are having comparatively greater size, and one can observe this cell with our normal eye. So you can observe this is egg. If you just boil that egg, you'll find that inside material is getting solidified. Actually, in case of egg, we are having a nucleus-like part which is yellow in color. We are calling that protein as globulin. Whereas outside part, which is actually oily, jelly-like oily part is there, we are calling that as albumin. But on heating above 52 degrees Celsius, this gets set and we are getting this white color part. We are calling that as proteins are set here. Okay, anyway, just imagine it's open egg, it is not boiled egg. Then you will find a fine membrane under hard calcareous shell. So we are having this fine membrane. That fine membrane I can consider here as plasma membrane. Now inside that there is nucleus. So here also we are having nucleus. Now nucleus is double walled. My drawing is not good, so I am showing here something like this. Actually this is double walled nucleus and there are pores. That means holes are there inside that. Nucleus really controls all the activities going on in the cell. Now this is nucleus. Now inside nucleus, if you observe, we are getting a material that is we are calling as chromosomes, but they are not visible throughout the time. But I am showing here some sorts of coiled structure. They are chromosomes present inside the nucleus. Now this is nucleus. From that these pores are there, and then we are getting some material like this. Even you can check out in egg also, you are getting something white material sticking to this part. This is something like this part. Folds are there. Membranous part containing folds. We are calling that as endoplasmic reticulum. So this is endoplasmic reticulum. This is nucleus. Now many times on this endoplasmic reticulum there are granules. So this is called as granular endoplasmic reticulum. Sometimes they are not having granules, they are having only smooth structure. So we are calling them as smooth endoplasmic reticulum. These granules are made up of ribosomes. So here these granules are made up of ribosomes and they are carrying out work, that is function, that is that is protein synthesis. So from amino acids, proteins are synthesized over here with help of these ribosomes present on this endoplasmic reticulum. Then we are having certain empty spaces inside. if you observe, they are having some bubble-like structure. We are calling them as vacuoles. So here, this type of vacuoles are there. If you boil out egg and just remove the upper part, you will get inside egg, but it is not having smooth shape. Usually it is having some curve at one end. This is because some vacuole is present here. So same way I am showing here, there is some presence of vacuole inside that. Then we are getting here another bodies. They are filled with enzymes. Enzymes are chemicals. They are carrying out or rather carrying out, I should use word, triggering out the chemical reaction. So some different 60 types of enzymes are present in this body. We are calling that as lysosome. I am revising the word. They are called as lysosomes. Then we are having certain discette shaped structures and some bubble-like appearance along with that. We are calling that as Golgi complex. So these type of structures are present. I am not showing entire all structures present. Again, keep in mind, all structures are not visible at a time in the cell. If you observe cell, let us take example, onion cell under microscope, you will observe only cell wall or box-like structure, bricks-like structure rather. Nothing will be there inside. If you add drop of iodine, tincture iodine. What is tincture iodine? Solid iodine is insoluble in water. So we have to dissolve solid iodine in alcohol and then it can dissolve in water. So this solution is called as tincture iodine. So if you add drop of tincture iodine, then only nucleus is visible. Like that, various substances we are adding out so that certain parts of cell are visible. We are calling that as stain. So here we are having some name of stain. I am saying iodine. Tincture iodine is one important stain we are using to observe nucleus. Then if you want to observe chromosomes, then chromosomes are absorbing out a stain that is called as acetocarmine. So with help of acetocarmine we can observe chromosome. Then eosine is another name of stain. So like that, various stains are used and then various parts are observed. Again, with help of ordinary microscope, there are different types of microscope. You are aware, simple microscope consisting of only one lens. Compound microscope is consisting of two lenses. Whereas their magnification power is different. But very high resolution. Resolution means ability to separate out things. Very high resolution microscopes are now available. We are calling that as electron microscope. With help of cathode rays, these type of observations are possible. So it is not microscope that we have to observe through that. We are getting display on the screen. So that is electron microscope. With help of that, we can assign different parts or we can observe different parts in the cell. So these are called as cell organelles. So basically, I am showing here only few organelles. This is not entire material inside cell I am showing here. Now, here I must compare with this cell. Just observe here, no organelles I am able to show. But here I can show various organelles present. So this is basic difference. This is membrane bound organelles. This is not membrane bound organelle. Now, if I want to tell you about plant cell, then plant cells are also having simple, similar structure like this. But certain changes are there. Say for example, plant cell are having somewhat fixed shape. Animal cells are not having fixed shape. So this way we are having a cell wall. The material used for cell wall, that is called as cellulose. So here, basic difference between animal cell and plant cell, that plant cells are having cellulose cell wall around it, whereas animal cells, they don't have cellulose cell wall around it. Plant cells, animal cells are not having any fixed shape, but plant cells are usually rigid. They possess a simpler fixed shape. Now, nucleus, it is there as it is, same to same. I am not drawing entire everything. But what is the more prominent thing in plant cell? That is the vacuole. Here we are having number of vacuoles, but size of vacuoles are small. Number of vacuoles are more. But here we are having large vacuole. Vacuoles are empty spaces, membrane bound empty spaces. Usually certain functions in cell, they are carried out in vacuole. Let me explain. Suppose certain foreign particle is there, it is trapped in vacuole. Then these lysosomes get attached to vacuole. Out of 60 enzymes, required enzymes are secreted here. That particle is dissolved. If it is food particle, it is dissolved, digested, that we are calling as breakdown is carried out, and then it is absorbed here. The waste material is there inside this vacuole, it is thrown away. So this way vacuole functions here. This is the function of vacuole. In case of plant cell, there is no permanent excretory system. So waste material is stored in this vacuole, and so we are having large vacuole. Here we are having another thing that I have to show, that is called as chloroplast. A green color pigment that is appearing here, we are calling that as chloroplast, which is not present in in animal cell except Euglena and Chlorella. Apart from that, all animal cells, they don't have chloroplast, but plant cell, they have chloroplast. So like that, basic differences I am discussing here. There are many differences. Say for example, at time of cell division, we can observe something, it is present, it is not present, like that. But right now we are functioning, we are focusing on our function purpose. So here I am explaining basic differences. Cellulose cell wall present, absent, fixed shape, not fixed shape. Chloroplast are present, chloroplast are absent except Euglena and Chlorella. Vacuoles are less in number but larger in size. Vacuoles are small in size but larger in number. So this is basic differences. Now you have clear-cut idea what is prokaryotic cell and eukaryotic cell.